welcome everybody to this webinar. It's great to see you. If you could just drop me a little comment in either the Q&A or the chat, just to let me know that you can hear us okay, that you can see everything, that all the technical stuff is working as we expect, and then we can kick off the webinar. So I'm just going to go over and check here. Looking good, looking good. Sounding good. Fantastic. Any questions you might have during the webinar, you can just drop them in there and we'll get to them in a little Q&A at the end. But until then, it is my great pleasure to welcome all of you and our very own Dr. Kasper Kragh to the webinar platform. Kasper has given a lot of webinars, is an excellent speaker and an expert in this topic that is going to be talking about today, and it's something we are extremely excited to share with you. So without further ado, wound in a cup, over to you, Kasper.
Thank you so much, Ben. And I'm very happy for all of you joining. As Ben says, this is something that we are really excited to work on, especially because it is something that we actually think that we can contribute a lot to as SimCell, but also because we are really interested in working generally with biofilm infections, and we have been working for several years with implant infections and cystic fibrosis infections, and this is really one of the ones that we're missing out on, chronic wounds. So through this talk, I'm gonna first start out talking a bit about chronic wounds in general and some of the stuff that we know, and I was gonna share some pictures from my academic career and before and from other researchers for us to really put a baseline on what are we actually looking at when we're looking at chronic wounds. And then I'm going to talk a bit about how we develop this model system that we're working on and some of the thoughts that we put into it. And then I'm going to show you a bit of data on some comparisons to some more simple models. So, but let's first start to just talk a little bit about like how to model a chronic wound infection, because as I write here, it's a messy affair. And it is really, if you look into what is a biofilm infection, we've done a lot of microscopy over the years of these infections and taking out sampling and actually looking into it. And many people, when they think about a biofilm, they think about something that's attached to a surface and thick biofilms that in some of our model systems. But when we actually look into the infections itself, it's actually a bit different, the picture that we're getting. It's actually more aggregates that are spread out inside of the infections. And as you see here from a cystic fibrosis patient, here are these aggregates, or here in a chronic wound from Kragh, and here from OTS Media, it's quite often these very small aggregates. And it is rare that they're actually sitting directly on a surface as they are here in the osteomyelitis, but they are more floating around in inflammation. And here you see the red here, that's bacteria. All the blue here are eukaryotic cells and primarily immune cells that are trying to attack these aggregates. And this is the same thing we see in more or less all infections that we have been looking into, especially when there's soft tissue involved. This here is a piece of debridement and pictures from my old colleague Les Quick and from his paper from 2022. 20, and this here is a debridement, so shavings from a chronic wound. And here is the whole piece of tissue that's been grained off from the wound itself. But if we zoom in this little area here, very closely, then you would actually see all these blue dots. That's immune cells. And down here, we actually see the bacteria. We zoom in a bit on that. Here again, the red are the bacterial cells in small aggregates, completely surrounded by immune cells. But in this very messy system where you have, this is quite deep in the wound, we quite often see them lying on the edge of the wound bed itself, but sometimes they can also be higher. And depending on which species it is, they can be in the zones that are very rich in oxygen, the very oxic zones, but we also see them in the anoxic zone. So it's a very complex environment. So when we are thinking about a chronic wound, we should really get out of our head the thick biofilm sitting on a surface. It is instead, when we find the bacteria, it's back in biofilm aggregates. It's never planktonic, very rarely planktonic, and it never sits on the surface. And then you really have this huge influx of immune cells. One thing we have been working on for a long time, understanding some of these infections, both in chronic wounds, but also in generally with chronic biofilm infections, is really how the microenvironment is really impacting these biofilms. And as you see, this mess of a infection is filled with so many different layers and a very diverse microenvironment. You have oxic zones, you have anoxic zones, you have a very complex nutrient environment. You have gradients coming towards the biofilm and even within the biofilms itself. We've always talked a lot about the biofilm sitting on a surface. We really understand a lot about these mushroom structures and a lot of things, the way have a liquid flowing over the surface. But that we have to get away from that thought of how thinking about biofilm when it comes to infections. It's much more of these aggregates. So the reason why we are really interested in this microenvironment and how it's actually impacting the bacteria, it is because when you have this microenvironment, this will affect the metabolism and the phenotype of the bacteria in these aggregates. And this is really what drives a lot of the tolerance against some of the treatments that we are applying for these infections. And now I think I said aggregates like 10 times already, but the microenvironment, the aggregates, and the media, the area around it, is really a prime driver for tolerance. When you think about these biofilm aggregates, you will have gradients coming towards them. That is how oxygen, how nutrients are accessing the bacteria, but also inside of them. This create very diverse metabolic activity, even inside of the aggregates, but also between the aggregates. And we know that most antimicrobials are very dependent on the metabolism of the cells. So in most cases, a dormant or very slow metabolic active population will be much more tolerant than the cells that are fast growing. So we need to take that into account that the bacteria, when we want to test a treatment on it, it actually has to be in the state of that some of these aggregates are in the infection. So, and that's will kind of lead me to saying, okay, we haven't, up until now, we haven't fixed this whole biofilm problem. Why haven't we found a really good treatment against biofilms? And this is, of course, the same in most of the biofilm infections. But of course, here's a bit of focus on the wounds, chronic wounds. And it's really not a single answer. There's a sum of many things. One thing is that in many cases, there has been a lot of focus on testing against the wrong state of bacteria. You cannot work with fast-growing or planktonic cells if you want to mimic a biofilm in the lab and really test a new treatment on it. And we really also need to think about the real conditions in a wound or in other infections to mimic that when we are developing new drugs, new treatments. If you want to test a dressing, a new compound on a biofilm, it cannot be something that's just fresh running on an agar plate. We have a tendency as scientists to kind of go into a very simple or reductionistic model system. And it is really nice that you kind of control the whole thing and have a, a minimal media or LB media running over a surface in these lab models. And I love a good old microtiter assay, but there is very little plastic surfaces and LB media in a chronic wound. Then quite often we have a tendency to also do it in very high oxic environments where you have a lot of aerobic growth. And that is rarely the case in most of these infections because we quite often have the bacteria very deep in the wound. So quite often it will be microaphilic to anoxic environment. And then again, we need to think about how we actually can model the media conditions because it really impacts how the bacteria grow and if they are metabolic fast, slow, or which genes it is they are expressing. So all of this leads to misleading results that then can lead to clinical failure of a product. One thing I also want to talk a bit about is detection bias. We have a tendency to always go towards CVU counts because that's the standard. It's quite often what we base our um, regulatory recommendations on. But it is actually CVU counts are quite flawed when as unsuitable when it comes to biofilm research. I'll come back to why this is. Of course, then there's also been a lot of work done with microscopy. I have myself practiced a lot of live death staining over the years, but live death staining actually have a tendency to, if you want to build a biofilm and then apply live death staining to test a compound or treatment, it actually overestimate the effect of the treatment quite a lot. And that is actually in the biofilm because quite a lot of the cells are overproducing eDNAs that will be surrounding the cells, and they will actually start lighting up red if they are treated with propionimidine, even though they are still viable and could actually still grow. I want to just go a bit deeper into why CFU counts sucks so much. And this is really a CFU count when you're working with biofilm has a lot of limitations. It is we want to remove cells from the biofilm that we have treated, and then we want to shake them up with sonications or vortexing, and then we want to plate them onto a agar plate. And one of the things that we quite often use is sonication. And we expect that sonication will break up these aggregates or these biofilms, and then it'll be 100% homogeneous single cell suspension. It does not. I've looked through many, many sonicated cultures, and it will still be filled with aggregates. It will break aggregates up into smaller aggregates, but it will still have aggregates inside of it. It's very hard to remove all cells into a planktonic state again. This also means that you'll have aggregates that will land on your plate when you're trying to count CFUs. An aggregate that is 10,000 cells will still come up as one colony, as well as one single cell would do. Then there's also the problem of taking cells that you remove from a biofilm, from this anoxic environment where they belong with low metabolism, and you pull them out, and then you put them on a super rich oxic environment, place them on an agar plate based on LB or Müller-Hinton. There's a lot of cells that won't be able to survive that treat. They will die of oxic shock. Even though that is anaerobic, facultative anaerobes and can survive most of it, it will be really hard. Then the treatment that you have tested could actually induce viable but non-cultural subpopulations in the biofilm, which is something we know and they will not be able to grow on a agar plate as well. So I always think it's a bit weird that we want to test things in a biofilm, but to test things in a biofilm, we remove the cells from the biofilm, and actually the only thing we're testing is who can grow on an agar plate as single cells. So if I had to make a list of a chronic wound model based on some of these ideas that I tried to present, I wanted to say, I want to test something that's clinical relevant. I want to test clinical relevant strains of bacteria. And then I want to have them in a state as non-attached biofilm aggregates. They should be floating in some kind of matrix, secondary matrix, and should mimic the wound condition by being complex, and it should be diverse, and we should have gradients through it. And we should try to add in so much complexity as the real wound have. Then I would love to get around this CFU counting and instead analyze the bacteria right where they are in the biofilm, in their natural environment. And we want to be able to detect these subpopulations that we know are lying there. that might not be able to grow on an agar plate, but still is viable. It would be nice if it was kind of high throughput, so you could test a lot of things at once. But again, there's really something beautiful with a 96 well plate where you just like spam out samples. Then I would love to include a immune response or immune cells into the mix, be able to test multi-species infections, and really to see this phenotypic. I want to create model system where bacteria are as they are in the natural environment. In the old days, I always heard Marvin Whiteley say, like, why are we testing? Why are we testing the what is it? That testing the bacteria in a liquid culture instead of looking at it as they are in a real biofilm models is like testing the I can't remember, but it was something with the ice bear or something. Okay. So we want to get better prediction so we can create better treatments. And we can do that by using better model system. So so some of the considerations that we just came through, we have tried to incorporate that into some new models that we have developed at SymCell. And here we really want to marry microcalorimetry with a model system that can really add on some complexity. And I think that a lot of you probably already know what microcalorimetry is, but just to recap, if you don't know, microcalorimetry is the measurement of heat being produced through metabolic processes by living organisms. So if you have cells that is yeast cells, bacterial cells, anything that is alive, they will produce heat by just having basic metabolism. Through our platform, you can then measure this in real time. And the neat thing about this is that this works really well if you have complex model systems, because as we're measuring heat, it doesn't matter where in our sample vials you are. So with the microcalorimetry, you're able to measure a real-time output of the metabolism. So saying like whatever's going on in my sample right now. And it measures the total metabolism. So it also have like slow growing cells that doesn't have to be fast growing or anything. It will still be able to measure that metabolism. And we can do that without disturbing the sample. So we can keep everything in place and be able to see if you add on a treatment, how would this actually affect a sample. So this works really well, as I said, in some of these complex model systems. We have been working with a lot of biofilms over the year, from environmental to infectious model systems. And the neat thing is that in a biofilm, we can have cells that are deep inside of the biofilm that we will still be able to detect the activity from with microcalorimetry. And this is really, when you look at some of these aggregates that we want to build in our model system, we are not really interested in killing one log of the biofilm or two logs. We want to detect like what is surviving like deep inside of these aggregates in the infection model. So the very, very last 0.0% of the population. And based on this metabolic output that we can have over time here, we are actually able to see, okay, are we actually killing the biofilm? Are we killing the cells in it? Can we detect these small subpopulations that are just surviving? But we can also see if there's actually more of a bacteriostatic effect. Are we actually limiting the growth of the cells as we build them, as they are being treated? So we have mixed this technology with something that was developed by Marvin Whiteley and his company SynthBio in Atlanta. They call it the chronic wound media 2. And this is really a media that has been developed to tackle a couple of problems. That if you put bacteria into a laboratory media, LB media, minimal media, Müller-Hinton, the bacteria will be in a completely different state than they would be in a chronic infection. And this will both be transcriptionally very different and also metabolic very different than in the infection. And as we know that some of these phenotypic factors are super important for the buildup of tolerance and resistance in some of these infections, it is super important for us to actually get closer to the real state of bacteria in infections. So Marvin and his team has been working on using RNA sequencing data to actually build media that mimics the real wound. So they have collected vast amount of RNA-seq data from chronic wounds from around the world. And based on this data, they have started to compare different types of media and actually seen how the bacteria are not at all behaving as they should be in some of these model systems. But then based on that, it's made small adjustment and designed a media that will actually ensure that the bacteria are as close as possible in a state as they are in a real wound. And really expressing some of these tolerance and resistance factors. So we have bacteria that are in a biofilm state inside of this model system, but in a way that we can test treatments on it and actually mimic what's happening in the wound. So what we have done is that we have then taken their media and then applied into, we have these small plastic cups that are inside of our titanium vials. And then we mixed in their media with a planktonic bacteria, just in this case, Pseudomonas aeruginosa, I'll show you. And there's viscosity in the media that will immobilize some of these cells. And over 24 hours, they will develop into a small wound where you have this, based on this media that has this very defined wound characteristics, these aggregates will develop. What you then have is a chronic wound in a cup, as I say, that you can then apply different treatments on top of it. You can add on dressings, you can add on lotions or whatever you want to test on it. And you will have diffusion of the treatment down through this little cup. But you will also have oxygen diffusion from the top. So you'll have zones where you actually will have in the bottom will be more anaerobic and or anoxic, and then towards the more anoxic zones. So you have bacteria in different states, but also inside of these aggregates, you will also have these diffusion ratios. So it's a way to kind of having a, a small wound in a cup. Have a very grimy picture here of it. You can barely see some of the aggregates lying around here in this. So we wanted to test this model system and say, like, okay, is this actually more closer to reality than that if you take a simple model system? So we wanted to test it against something that would be quite simple. And we took a LB media and added a 0.5% agar that will give, it will mimic the viscosity of the wound media. And also create these nice biofilm aggregates, but based on a LB media. So you don't have to go through it, but just I want to show that this is a very simple experimental setup. You mix in bacteria, you put on a lid, you grow them, and then after 24 hours, you can add on a treatment. In this case, we took and actually wanted to look with Pseudomonas aeruginosa. So we used a simple PA01 strain, laboratory strain, and then we took a isolate from a wound that we got from Thomas Bjørnshol's group in Copenhagen, uh, and grew them for 24 hours. Then, as a test experiment, we just added different concentrations of ciprofloxacin in this case, but you could exchange that to any other kind of treatment. So, if we just take a look at what happened then during the first 24 hours of developing these two strains in either the simple LP media or in the wound media. And what you'll see here is that we have this development of both the PA01 strain and the clinical isolates growing really fast. So this is the metabolic activity over time. You'll see that this big peak, and then they go into a bit of a steady state. It goes much slower when looking into the clinical isolates and the PA01 in this wound media. So everything is much slower, but over time, they actually uh, produce the same aggregates. But just looking at the maximum height, so the maximum metabolic activity, you can actually see that the LP-AGA is much higher in activity than in the wound media. But also notice that the clinical isolates actually peaks out much lower than the PL1, which is not that strange. We know that they have quite often been adapted to the wound. So what happens then if we apply a treatment on top of it? This is the PaO1. And PaO1 here in this case in LP-AGA, and we apply then 0 and 0, 0.0, zero point 0.5, and 1. This is around 10 times MIC, and this is 1 MIC for this strain. So what you'll see here is that we actually see a lowering of the maximum metabolic activity and over time. And I know it's over time within the first. So this is on the fully developed aggregates. So the wound that is already mature. Well, let's compare this then to what happened in the wound media. And what you're maybe able to notice is that not that much happened in the wound media to the strain. The effect on the aggregates are much lower. And the same thing is true with the clinical isolates. You also see that the effect of the treatment is much lower. And if we then compare that, so here, this is the clinical, the PL1 in the wound media and how it actually is affected by the treatment, whereas in the LP-AGA, it plummets down here. And not in the same rate, but it's still definitely the same significant difference in the clinical isolates. And if we look into just saying, looking at the reduction in max metabolic activity here in the wound media, it's around 18%, where we then have a reduction of 63% in LB-AGA, and we have about the same reductions here. So if you just took your treatment and tested it on an LB model system like this, it actually worked really well. You had a good reduction, and you could actually maybe be quite positive of this. But if we actually put it into a real clinical or a more clinical relevant model system, we are actually able to distinguish the fall in metabolism actually much lower. So, and if we then look into and say just the total metabolism, how affected are these aggregates if you don't put any treatment on them and just say how much energy are they output? It's actually more or less the same. It's just the rate for them to get there that is very different. But the treatment really paints a different picture. So if we just take a look at this outcome in this model, this was same treatments, same strains, and all in biofilm aggregates. But the difference is really that if you looked at the LP-AGA, you had a severe bacteriostatic effect, a really effect on it, where the wound media actually had a bit more of a limiting effect. So it's really here where we say, like, if you put there's much higher tolerance if you put it into more realistic conditions by using this wound model. And there's definitely also some strain-specific effects on clinical isolates. So the clinical strain was a bit more tolerant in LB media, but we definitely still saw the difference, but limiting effect in the wound media. So I would always argue to using clinical isolates instead of a biofilm, instead of a lab strain. So we have done this with other biofilm infections. We have also worked on a sputum, cystic fibrosis sputum model system where we wanted to look into, can we make a cystic fibrosis model. And here we added on much more aggressive treatments compared to what we had here in the wound. Here we used tobramycin, but else things was done the same thing again with a Pseudomonas aeruginosa, and then compared to a LB agar. And in LB agar here, you will actually see that we are getting more or less close to eradication. At least we get a very, very intense effect of the treatment with tobramycin compared to the untreated. But if you then compare to the synthetic sputum media, then we can see that we actually have we definitely have an effect. But this here continued metabolism, even at very high concentrations. So you have here some of the subpopulations we still are able to detect. So very much the same lesson, like how you develop these models really determines the outcome. And you could again have said, this worked great in your LB model. So just to kind of wrap it up a bit and saying like, if you take this model, we have actually think that we have something that could be quite interesting to work with and develop further on. This is small scale, very reproducible wound models, and enables this like real time investigation of this very complex model system where you have some of these aggregation, you have the diffusion, you have some of the nutrients that actually make the bacteria behave the correct way. And in the system, it's actually able to run 32 small wounds at a time. It might not be a 96 well played, but it still gets a job done. And it takes away this CFU part of it. We can test it right where they are without removing it from the model. And it can enable us to really detect some of these subpopulation and some of these other effects than just a very binary, can you grow on an agar plate or can you not grow on an agar plate? So it's much more dynamic data. There's still a lot of things that could be expanded on here. We could add a immune component. If you added in isolated immune cells to the wounds, that would be very interesting to see the effect on that. They are a big part of developing some of these gradients that are in wounds and in chronic infections. There could be a possibility of doing multi-species infections, of blending different bacteria and looking at synergy. And of course, could also be really interesting to go further on this, like different strain-specific dynamics. If you actually have a real resistant bacteria, multi-resistant bacteria, how would that correlate together with the tolerance that the biofilms really give the bacteria? So just saying that chronic wounds and LB plates is not my favorite blend. So you probably understood that from now. But if we actually add in biofilm aggregates, different anoxic and oxic zone, a complex nutrient environment, if we can add in immune response, this really moves us very, very closer to the real wound. And it could really be a way of jumping over some of the wasted resources in developing something that might not even work in the real patient. So there's a very large tendency to overestimate the effects from standard model system. If it's drip flow reactors, if it's microtiter assay or CDC reactors, those biofilms are not representative of the ones we see in patients. So we think that this model system, this pairing of microcalorimetry and wound media, actually provide a really nice real-time readout of the metabolism and can really give a clinical relevance to model systems. And as you saw, we could definitely see how the difference between LB and these clinical or this biofilm media really have a big difference. So I think what I really want to brings us a take home message is really let's make better models so we can make better predictions and better therapy for the patients. It is really you have to be messy when you make biofilm infection models because they are inherently messy. So this whole thing has been really carried out a lot by Emma and Matle and Costantina's in the lab in SimCell, but with a lot of good input from Marlon Whiteley at SynthBio. And then I used a lot of old data from some of my old friends at University of Copenhagen. So by this, I will like to say we are really interested in talking more about wounds with anybody who wants to discuss it with us. So please to reach out, and then Ben, I will send it over to you.
Thank you very much, Casper. Yeah, thank you everyone for watching along and listening. I know some of you had to go, but please take a note of these email addresses here in the website so you can go and visit. We've got more resources on the wound model. We've got an application note that maybe you have or haven't seen, but we'd be happy to send that on. So definitely please get in touch. And the recording will be available for all of you as well. So without further ado, we can take some questions which are coming in here. So, Casper, are you aware of any studies that identify population changes that could occur over time in the chronic wound, similar to studies that have been done in cystic fibrosis, and any kind of changes, basic knowledge or kind of other developments?
Yes, I'm pretty sure that was it Matthew Malone from Australia that did a study where they actually looked into uh, not as big a study as some of the cystic fibrosis development over years, but they did a quite big analysis of genotypic analysis of wound isolates from around the world. It's a couple of years old, maybe 4 or 5 years old, but I think it was done with Matthew Malone and maybe Thomas Bjarnsholt. Yeah.
Cool. Thanks very much. And another question here, kind of harping back to what's been built on by Bill Costerton, Niels Hoyby, DC White. But the question here is like, can we look at single cell metabolic activity, or how would that look in the future? Because talking about subpopulations, obviously you kind of want to know the different metabolic activities and what a single cell actually contributes to the metabolic output. How far are we away from that, or can we do something about that now?
That is very interesting, and I definitely I think there's a developing school of thought in biofilm research that we can still get what we define as biofilm as a biofilm phenotype without it being built into a thick layer of biofilm. And the question is, can you actually be one or two or three cells and actually have, because of your metabolic state, to be in a biofilm mode? So you actually will have everything that we normally will list with the characteristic tolerance but it is something we definitely need to do much more research on because to build something that is in a dormant state might not be enough because that we know that there are internal regulatory things so you will need to have a upregulation of cyclic GMP and several other things to express some of these biofilm characteristics. But could you still be protected against antibiotics? Could you still be resilient against the immune response by being in a single cell state, but just be dormant? I think that's very interesting, and I think it's very timely to start looking into that. Yeah.
Cool. So you're not the only person we've had on the webinar platform here to kind of rail against CFUs. Willem van Wammel did a very, very good webinar, and I recommend that everyone listening go and look at that. Can we get rid of CFUs already, or do you still think there's a place for them, or how do we use them going forward, given all the regulatory aspects relating to CFU?
It is a huge battle that we need to take as microbiologists, especially as biofilm researchers, because it is all regulatory guidelines that are bound up on this, and it is very problematic. as we there has not been presented anything good alternative that has been accepted by the regulatory bodies and the regulars. And of course, I would of course always say that uh, microcalorimetry would be the biggest uh, uh, fact or possibility there. But we know that to move regulatory bodies and regulatory guidelines, it takes decades, and we have tried to shout this for a very long time. Right now, we have a fantastic cost action going in the EU, the regulatory toolbox that is actually focusing in on how can we actually move some of the regular areas, both in EU but also in the US, towards accepting alternative than just CFU, because it is a deeply flawed system. It has worked a lot for a lot of good things, but we need to move on from it.
Mm. So familiar, isn't it? It's just so easy to go and do a plate count experiment, but it's really cool to see all these models now. And I think specifically with this one, that as long as you have the calScreener, it's pretty straightforward to do. It's not like you need really a very, very complex model, even though the result is very, very similar to what you'd see in a wound. So that's nice. Um, so what are we looking at here in terms of benefit for the patient and new drugs? Like, are we going to use this model and we're going to look for brand new compounds, do you think? Or could there be some kind of easy wins where we've got stuff on the shelf that's already kind of approved and that maybe in combination we could actually look at some
Yes,
really, really effective treatments that are kind of just waiting to be used. We just need to know the right combinations.
that is definitely like, because we know how slow things move when it comes to developing a new antimicrobials. Of course, when we're talking a wound care, it is easier to treat. It's easier to get approval of treatments than it is when you have oral treatments. But it is still something that moves quite slow. But I think specifically what you say there, can we develop synergistic treatments where we already have stuff that are approved and mixing them together? That could really be where the easy wins are just there. So we know that you can very easily, not very easily, but you can when you're attacking a bacteria or biofilm with different fronts, if we are working with a phage treatment or we have a chemical treatments of enzymatic solution of the biofilm matrix, and you then hit that together with antibiotics, then you have fantastic effects. Some of the stuff that Ken Aruba from La Roque University has done with enzymatic treatments of wounds in her mouse model is very, very exciting, and that definitely has a lot of potential. But it's also very clear that a enzymatic treatment that just loosens the bacteria from the biofilm doesn't work alone. You need to have it in combination with other treatments. So yes, let's see what we have on the shelf and try that first. It's much easier when you have two approved drugs and just mixing them together.
Cool. So we talked a little bit here, and the questions about, so we've got the chronic wounds, we've got cystic fibrosis. Like, are there any other applications where you think this model and this way of looking at bacteria is going to be particularly effective?
I think it's definitely something where you can think about other treatments where have very complex and messy treatment infections. So synol fluid around implant infections that come to mind as one of the things that we have a very hard time understanding, and we know that we have floating aggregates there sometimes not there. And how can we actually add that in? There's other things where you have a soft tissue and you have a what was I about to say? Um, but as long as you have something where you can add on the complexity, and maybe you'll even have a we have a customers that are working with dental understanding dental infections, and is there something where we can actually add on the complexity of saliva in the mouth and still having a non-soft tissue as the tooth and build up different zones. There's so many things that you can build of really exciting model systems that doesn't have to be impossible to do, because if you build some of these model systems and are relying on microscopy, it starts to become. Uh, I would say that one of the things that just came out of paper a couple a month ago from Oliver Basset from University of Basel, that made this fantastic model where they had agar plots and had these implant screws, and actually looked at the colonization with different coatings on these implant screws, where it's both complex and very simple at once. I think that's excellent. Yeah.
Yeah. And we also have the webinar there from Olivia. He gave a webinar
Yes.
just after he published. That's also in our library if you'd like to check it out. So another question, a brief observation. So the number of applications in non-medical fields, given the economic impact of biofilms in fouling, corrosion, and the positive impact of biodegradation, do you feel, Kasper, like there's things to explore there with microcalorimetry?
Very much so. That's a fantastic question. So it's a really question, an observation. I think that could be a huge opportunity, also because we're again talking about a complex environment. I joined a workshop not long ago in London for microbial induced corrosion. That's a huge problem where, again, it's biofilm on a complex surfaces in pipes. We need to understand the interaction between the surface and the biofilm there. Could we actually even measure corrosion as a chemical reaction besides the bacterial metabolism? Um, I think there's a huge potential to using microcalorimetry there, and that would be a super exciting application. Now I just railed against surfaces, but there we kind of need a abiotic surface. Um, but we have also customers that has working with stuff like soil and wastewater, where bioremediation is really key to how they can take up where the biofilm is a positive thing. So that could definitely also be a really exciting opportunity.
Cool. Well, I don't see any more questions, so I'd just like to take this opportunity again to thank you all for coming. The replay will be available if you'd like to watch again or send it to your colleagues. We have more resources, so please look out in your emails and reach out to us here if you have any questions or you'd like to learn more about some of the experiments we could do. Thank you again, Kasper. Always a pleasure. Very insightful, very entertaining, and we look forward to the next one. So thank you, everyone. See you next time.